Hey everybody, in this video we are going to transition to the RNA mechanisms part of this course. So most of the stuff we have been talking about in the previous lectures deal with DNA mechanisms, and we sort of transition into RNA by going through transposons and viruses. Some of our viruses, the adeno-associated virus was a DNA-based virus, and HIV was an RNA-based virus. And now we are really going to focus in on RNA mechanisms and different types of RNA in mostly eukaryotic cells. So because we are talking about RNA molecules in eukaryotic cells, we need to first introduce the various types of RNA polymerases in eukaryotic cells. So we have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And some types of eukaryotes have additional RNA polymerases, but all of them have these three, and these are the primary RNA polymerases. And eventually, I will transition to calling these just pol 1, pol 2, and pol 3. Now, the primary RNA types transcribed by each of these RNA polymerases are, let's say for RNA polymerase 1, that would be most of the ribosomal RNAs. So these would be the 18 Svedberg, the 5.8 Svedberg, and the 28 Svedberg ribosomal RNAs. So these are the RNAs that are part of the ribosome. And these are the ones that are found in the ribosomal RNA repeat arrays on the acrocentric chromosomes of humans. So that'd be chromosome 13, 14, 15, 21, and 22. Maybe you remember that from an earlier part of the course when we were talking about the ribosomal DNA arrays on those chromosomes. Now, RNA polymerase 2 is going to be the polymerase we focus on mostly because it's in charge of transcribing messenger RNAs. And it also has a role in some other types of RNAs, small nucleolar RNAs, microRNAs, and some other types. But the main one we'll focus on is messenger RNAs. So RNA polymerase 2 is the only one that transcribes RNAs that are translated into proteins. RNA polymerase 3, its primary job is to transcribe the tRNAs. So those RNAs that transport the amino acids to the ribosomes during translation. Now it also transcribes the 5 Svedberg ribosomal RNA. Now Svedberg, if you forget, that's just a size that is used sometimes for different biological molecules. Now the 5S repeat array is found on chromosome 1 in humans. So pol 1 is in charge of most of the ribosomal RNAs, those that are found on chromosomes 13, 14, 15, 21, and 22. Pol 3 is in charge of the 5S RNAs that are found on chromosome 1 in humans, encoded by chromosome 1 in humans. Now, I just want to touch a little bit on the structure of RNA polymerase 2 in comparison to the other polymerases. Now, the structures are very similar between these. And why are we talking about the structure? This isn't biochemistry, right? This isn't a course on biochemistry, but it's somewhat useful to get an idea of how big these proteins are, how complicated they are. So we're just going to take a look at the different components, let's say, or different subunits of these RNA polymerases. So RNA polymerase 2 has a 10 subunit core, and all of the subunits have named like this, RPB1. I'm going to go out of order here, and I'll explain why in a moment. So 1, 2, 3, 9, and 11. So that's 5 of the subunits of the 10 subunit core. And then we have RPB 5, 6, 8, 10, 12. Now these are all protein subunits. And interestingly, so each of these, pol 1, pol 2, and pol 3, all have 10 subunit cores. So 10 proteins that make up the core of these polymerase holoenzymes, and we call it a holoenzyme when more than one protein is involved in making the functional enzyme. So these same proteins, RPB 5, 6, 8, 10, and 12 down here, are also found in pol 3 and pol 1. However, the first 5 are different in pol 1 and pol 3. So there's an RPB1-like protein in pol 3, 2-like, 3-like, so not encoded by the same gene, but overall the structure is very similar. So same thing for pol 1. And you know, we can probably imagine that these have to be somewhat different, right? Because they have to interact with different general transcription factors to make sure they're transcribing the correct genes. And it would be interesting to see how these structures, you know, what the differences in the structures, how do these differences allow them, say, to transcribe their particular genes better than others. Like for instance, messenger RNAs are often much longer than transfer RNAs. So is there something about the pol 2 structure that optimizes it for transcribing those long messages compared to the short transcripts transcribed by RNA polymerase 3? I don't really know. I'm sure there is something important in there. It's more of a biochemistry question, isn't it? So in addition to the 10 subunit core, there are some, we can call these peripheral subunits. So we'll just talk about this one. So RPB4 and RPB7, and that's in RNA polymerase 2. So RNA polymerase 2 has a 10 subunit core and a 2 subunit peripheral complex. Now, okay, what does this structure look like? I pre-diagrammed the structure. This is based off of something in the notes, or a diagram in the notes. So make sure you check that out. So let's see if this works. I'm going to select this down here, and I'm going to bring it back up top. Here we are. Okay, so if you took a look at the notes, you've probably seen this already. And what it is supposed to be, it's supposed to be a cross section, like if we had RNA polymerase 2. Now this would be a cross section of the polymerase, and this is a polymerase. This is pol 2 in, you know, transcribing a nascent RNA. So nascent, I need to go back to my paintbrush here. So a nascent RNA is an RNA that is still undergoing transcription. So this red thing right there, that's supposed to be the nascent RNA. This would be the 5' end over here. Now the 3' end, let me zoom in a little bit. 3' end is in here. So that little pink dot in the middle, that represents a magnesium cation that is involved in transcription. So let me find a better color here. Well, maybe that's a better color. So, okay, so we're looking at RNA polymerase II in the process of transcription. Now, researchers who study the structure of POL2 have named some of these things here. So we're going to call this the wall. So they've given names to the various parts of POL2. So we have the wall there. We have what's called the bridge down here. Up here is going to be the clamp. And you notice with this structure, it's a cross section, and the gray areas are supposed to be further back, so into the further away from us, the viewer who's looking at the diagram. And the lighter sections, these sort of eggshell colors, are supposed to be closer to us. And so this right here would be the jaw, where the DNA enters. We have the funnel right here. And the funnel is where the NTPs will enter so they can be added. So I'm going to put RNTPs, right? Because these aren't DNTPs, deoxyribonucleotide triphosphates. Let me put this over here. These are RNTPs. I keep hitting that button. I have to get used to this. RNTPs. So nucleoside, ribonucleoside triphosphates, right? Because we are transcribing RNA here. POL2 is transcribing RNA. So, and they will enter through the funnel, which is, sort of this big area down here. And this thing right here, this little narrow region, we're going to call that the pore. Now, let's see. In green here, we have the non-transcribed strand, or the non-template strand. We could probably call that the non-template strand or non-transcribed strand. In the blue strand right here, this is going to be the template strand or transcribed strand. Now, what else do we have here? Oh, we have another important component right here. So this little piece right here, we're going to call this the rudder. And okay, so what's going on here? So the clamp is supposed to probably close down over the DNA. And as the DNA is entering the RNA polymerase, it's already been opened. So the DNA is opened by a process that involves all of the general transcription factors, I'll say, and I'm going to get to that in a moment. So the DNA is unwound inside here, inside the POL2 complex, and we can see the template strand is being used with the active site, say down here, to add an NTP by complementary base pairing to the growing end of the nascent RNA. Now the rudder is supposed to separate or aids in separating the nascent RNA from the template strand after about 9 nucleotides. So let me put that in here. Or 9 base pairs, and the base pair would be sort of made up of DNA and RNA, right? It would be DNA-RNA hybrid, because it's a hybrid between the nascent RNA, a small section of the nascent RNA, and a small section of the DNA template. So separate DNA, RNA after 9 base pairs. Okay, and so after separation, the template strand comes out through, this is called the exit up here, and it can reanneal with the non-template strand, and the single-stranded nascent RNA exits also. Another interesting thing is this wall here. So because of the wall and the way the DNA enters the RNA polymerase, the DNA is said to make this sort of 90-degree angle in the RNA polymerase 2, and that may be important for orienting the DNA template inside the catalytic site. So the appropriate NTP can be added to the end of the nascent RNA. Okay, so that's it for the structure of RNA polymerase. Now let's take a look at the promoters. So we are not going to talk too much about pol 1 and pol 3 promoters. Maybe we do a little bit in the next lecture. This lecture, we're really, most of the details we go over will be pol 2 promoters. So pol 2 promoters. Let me zoom out a little bit. So my diagram here doesn't look so pixelated. So pol 2 promoters. Okay, so what is a promoter? So there are many ways to define a promoter. So let's just go with a short region of a gene that's needed to load RNA polymerase and start transcription. So short region of gene needed to load RNA polymerase, and this definition works for pol 1, pol 2, and pol 3, so I won't specify the polymerase here. So short region of a gene needed to load RNA polymerase and start transcription. So the promoters, essentially what we're going to focus in on is the core promoter. And so the definition of a promoter has somewhat changed over the years. So the promoters might include what we would call, some researchers would consider the core promoter, core promoter plus, let's say, these things called enhancer and silencer binding sites. Now, we're not going to talk about enhancers and silencers yet. Enhancers and silencers, so these would be sequences that sort of help increase the rate or decrease the rate of transcription, or maybe block transcription. The core promoter elements are in the core promoter, and these are just essential or critical for loading that RNA polymerase onto the DNA and to start transcription. So they're not accessory elements at all. They're not involved in increasing or decreasing the rate. We're considering these to be just the sequences that are needed to get that RNA polymerase into position. And so we're going to call that the core promoter, just that minimal sequence of DNA that's needed to get RNA polymerase into the right spot to start transcription. And then more broadly, promoter could include lots of other things that include elements that increase the rate or decrease the rate. So we're going to focus on the core promoter elements in this lecture. So let me diagram a promoter region for us. So we will start with a +1 site. Now that is a very important site. I have to decrease the sensitivity on my pen. I don't know if you guys can see that window popping up once in a while. It's really annoying. So, okay, so +1 site, that is going to be where transcription starts, at the +1 site. So the first nucleotide added to the RNA is added at the +1 site. So there are a couple sequences that are found in the core promoter regions of RNA polymerase II transcribed genes. So if we go over here, now I'm going to call this region right here the -36 to -26 region. So if we should go back for most genes, let's say in the human genome, from the +1 site and count backwards. So one thing I want to point out right here, there's no 0 site. So there's no 0 site. The first position before the +1 site is called -1. So if we should start at the +1 and count back 26 to 36 positions, we will very often, not always, but very often find what is called a Tata box. And Tata box has this consensus sequence. So TA, T, A, A, A. And that's 5' to 3' right there. Now I could put the complementary sequence in here, right? But we probably don't need to, because by now we know that in molecular biology it's very common just to show the sequence of one of the strands, because we can easily derive the other strand, the sequence of the other strand in our heads. So if we should look at most genes, let's say in the human genome or genome of another eukaryote, and we count back -36 to -26, look around there, that region, we should find a Tata box. And the Tata box usually has this sequence, TA, T, A, A, A. And sometimes we might see something like this, or maybe just A's. But most of the time, if we do a statistical analysis, we'll see something like that. And those are for genes with a Tata box. Some genes don't have a Tata box at all. But those that do usually have something like that. Now, okay, so that is a core promoter element. That's one example of a core promoter element. Another one occurs at the +1 site. We're going to call that the initiator region. INR for initiator region. Now the consensus sequence, if we should look at most genes in a genome, right around the +1 site, what we will see is something like this. We'll see a Y, 2 Ys, an A, an N, a W, and 2 more Ys. Now some of you are probably thinking, like, what is he talking about? Ys and Ws? That doesn't make any sense, right? So when we're talking about consensus sequences, researchers have come up with this sort of ambiguity coding system. So we can call it, let's say, consensus sequence ambiguity codes. And there are other areas where we can use this ambiguity coding system. But it's really useful for consensus sequences. Okay, and so what this tells us, and let me just talk briefly about how these codes work. So there's a bunch of these different codes. And let's say Y, what this is, Y stands for C or T. And it's derived from the word pyrimidine. This Y right here. So Y stands for C or T. And in the notes, I put a table of the ambiguity coding system. So N is any base, A, G, C, or T. W is used for any of the, say, weak bases. So bases that form 2 hydrogen bonds in a base pair. So what are those? A or T, right? A or T. S is for the strong bases. So G or C. Those form 3 hydrogen bonds in a base pair. Now, how about B? B is a neat one. So it is essentially means not A. So that could be what? C, G, or T. And D is another neat one. So it means not C. So that can be A, G, or T. So if we wanted to say, at say position 1 in a sequence, it can be an A, a G, or a T. Well, we could use D. So let's go back to the consensus. Here, let me, maybe I can, erase some of this quickly. Let's see here. I want to use that shape. Okay. Done. Okay, can erase that. Can erase this. Nope, didn't work. Sorry, still getting used to this new setup here. Okay, done. 2D shape. Done. Okay, so I erased all that. Okay, so we can go back to this initiator region consensus sequence. Now, okay, what was Y? So Y was C or T, right? C or T. So if we should look at most genes in a eukaryotic genome around the +1 site, well, we should see a C or T. This is going to be at -2. That's the -2 position. This Y right here, that's the -1 position. So we should see a C or T at -2 and -1 most of the time. This is the +1 position. So what this is telling us is that most genes most RNAs start with an A, right? Because A is found most often at the +1 site. So the +2 is any. There's no bias towards any of the 4 ribonucleotides, or DNA, I should say, deoxyribonucleotides, because we're talking about the DNA, the promoter element, not the RNA transcript. So W is A or T at the +3, and then we have 2 more G's or C's here, +4 and +5. So, okay, one more promoter element, and there are dozens of promoter elements, and we're only talking about 3 of them. And the reason why we're talking about these 3 is I wanted to show up that we can have promoter elements upstream, core promoter elements upstream of the +1 site, overlapping the +1 site, and downstream of the +1 site. Okay, so if we look at most genes and we look at +28 to +32, so around there, we might see some genes, maybe not most genes, but a lot of genes will have what's called the DPE. So downstream promoter element. Okay, and what is the consensus sequence there? For that one, the consensus sequence is R, G, W, Y, and V. And, okay, what was V? V essentially means not T. So it can be A, G, or C. And R is for purine, so A or G. Okay, so those are 3 core promoter elements. And, who cares? What are these things for? So the general transcription factors, which we will talk about next. General transcription factors bind the core promoter elements. So the types of core promoter elements, the number of core promoter elements, identity of the sequence of the core promoter element relative to the consensus for that element. These all influence how the general transcription factors bind to those promoters. And they influence, they influence what genes are transcribed in what cells. So to summarize, the core promoter elements are in charge or help initiate transcription of the genes by interacting or by serving as binding sites for the general transcription factors. Okay, so let's take a look at the general transcription factors. Okay. General transcription factors. Now these factors, these are the protein complexes. Some are individual proteins, some are complexes. So these are the guys that are needed to load RNA polymerases onto the promoter to start transcription. So they are not what we're going to call transcriptional activators. Which are the sort of enhancers and silencers, or proteins that bind to enhancers and silencers to sort of influence the rate of transcription. The general transcription factors are to just get that RNA polymerase into position and to initiate transcription. So let's consider transcriptional activators as proteins that influence the rate of transcription, and general transcription factors as, just the factors that are needed, critical to making transcription happen. Okay, so in eukaryotes, there are a number of transcription factors, general transcription factors, and I'll abbreviate these as GTFs. So we have TF2A. They have nice, easy names to remember, right? TF2A, B, D, E, F, and H. For some reason we skip C. There's no C, and there's no G. So some of these are very complicated complexes, no pun intended, with multiple subunits. Some are easier, so only 2 subunits for TF2A. Only 1 protein makes up TF2B. So D can be up to 15 subunits, so that's one of the more complicated one. E, 2 subunits, F, 2 subunits, and H, 10 subunits. And TF2H, maybe this rings a bell for some of you, because TF2H is a really neat complex, has 3 functions, right? One is going to be transcription initiation, which we're talking about now, but the other 2 are these things called TCNER and GGNER, and maybe you remember this. This was a couple of weeks ago. We talked about nucleotide excision repair. TF2H plays an important role in that process. So these will be the complexes we talk about when we talk about the general transcription factors and initiation of transcription. So, okay, so there are a number of complexes involved with initiation of transcription. So, to get a little organized here, initiation of transcription, this is what we're talking about now, which involves loading RNA polymerase II onto the core promoter of RNA polymerase II transcribed genes. And there will be a number of complexes involved here. We are going to first take a look at the upstream promoter complex. After that, we will see what is called the core PIC, which is the core pre-initiation complex. Then we're going to have the closed PIC. Now PIC is pre-initiation complex again. And then we're going to have the open PIC. So these names aren't critical. So different researchers come up with clever ways to name the different stages of how transcription is initiated, but we will use them because it helps organize the process. So, okay, what is the upstream promoter complex? Okay, what is that? So there are going to be 3 general transcription factors involved. So TF2D, TF2A, and TF2B. So first I want to talk just a little bit about TF2D. So TF2D is made up of a protein called TATA binding protein. And you can imagine this is something that binds to the TATA box if the core promoter has a TATA box. And a number of TAF proteins. So TAF stands for TBP associated factor. TAF stands for TBP associated factor. And so together, the TAFs and TBB, well, depending on what TAFs are part of the complex, it determines how well TF2D binds to the core promoter. So let's see, I can diagram this for you. So TBP is often diagrammed in a saddle shape, so it can sit down sort of on top of the TATA sequence. And then here we can have, they're up to 15, there are 14, I should say, different TAFs. And it's often diagrammed something like this. So we got all these TAFs here. And the TAFs can interact with the other core promoter elements, right? You have TBP interacting with the TATA box, but then you have all these TAFs here that can interact with the other promoter elements. So they can determine how easy it is for let's say TF2D to assemble on the core promoter by how well this complex or recognizes the different sequences in the core promoter. So I'm going to simplify this diagram, and I'll probably just do something like this. So this cloud-like structure behind TBP right here just represents the various TAFs that can be associated with TBP. So I already have this diagrammed for us to speed things up. So let me go down here and copy it. There is our RNA polymerase, and here is the upstream promoter complex. I'm going to select this, going to copy it. Go back up here. Okay, so what do I have here? Okay, so here are the TAFs that are part of TF2B. Here is TATA binding protein. Okay, so together those are TF2D. Here is TF2A, which binds to DNA at the core promoter, just upstream of TATA binding protein. And then here is TF2B. In TF2B, say the N-terminal region will bind polymerase 2, and the C-terminal region binds to the DNA. And when this complex assembles onto the core promoter, we get this bend in the DNA. So you can see we have that 90-degree bend. So that's kind of interesting, right? We have two 90-degree bends. Before we saw that there's a bend in the DNA that when it goes through RNA polymerase II, and we have another bend in the DNA in the upstream promoter complex. Oh. So this is the upstream promoter complex. And what does upstream mean? Upstream is all, it's sort of relative to the +1 site. And we have the +1 site diagrammed here with an arrow, the white arrow there. So that's the +1 site. But we don't have RNA polymerase here yet. So RNA polymerase is going to come in next. So the next complex to form is going to be called the core PIC. So the core pre-initiation complex. So that will involve TF2F and RNA pol 2. So TF2F binds polymerase 2 and helps position polymerase 2 on the promoter. And other roles and other interaction partners with TF2F are unclear, at least as of a couple of years ago. So let me copy that for us. Okay, I have that guy right here. You know, I can probably copy both. Oh, no, I can't. Select, copy. Okay. So this is going to be the core PIC. Why can't I find my pen here? Core PIC. So what we did was we added TF2F here. Again, we don't really know what else it's interacting with, so we have it just interacting with RNA polymerase II, and it's helping position RNA polymerase II onto the core promoter. And we can't really see the +1 site anymore. The +1 site's going to be in here somewhere. Okay, so the next stage, the next complex to form, is going to be called the closed PIC. So the closed pre-initiation complex, and that involves adding TF2E, so the general transcription factor TF2E, and TF2H. Now, TF2E binds immediately upstream of the +1 site, so right before it, and it binds pol 2. So TF2H, this one binds downstream of the +1 site. And it binds to TF2E. So binds both of these. Okay, so let's see that. Too far? This is the last structure. We're almost done here. Copy. Paste. Okay. So here we go. This is the closed PIC. Give me my pen back. Closed PIC. Now here is TF2E. Bound just upstream of the +1 site. And then here is TF2H. Now TF2H, this is that big protein that, okay, so 10 subunits. 7 form the core, 7 subunits in the core, and 3 are part of a kinase domain. And I think the kinase domain is going to be more important in coming lectures. It's involved in, I think, phosphorylating the C-terminal domain of RNA polymerase II. We'll talk about that later. But this is the kinase domain, and then this would be the core protein right here. So the darker, the brighter orange is the core of TF2H. And we can see it's interacting with TF2E, and it's bound to DNA upstream of the +1 site. Okay, and so and TF2H is really interesting. So it's thought to be the main, I should say, complex, the main complex involved in unwinding the DNA, so RNA polymerase can start transcription. The next complex is called the open PIC. So we're not adding any more subunits or complexes or proteins to this structure right here. The open PIC is formed when TF2H unwinds the DNA for RNA polymerase II. And once that DNA is unwound, then RNA polymerase II can initiate transcription. And it's thought to do the unwinding with its translocase activity. And, you know, helicases are things, are proteins that unwind DNA, and for many years it was thought that TF2H worked as a helicase, but now it's widely thought that it works as a translocase to unwind DNA at the +1 site for RNA polymerase II. And how does that work? So essentially, the DNA is held in place by like all of these things over here. So the DNA is like tethered, it can't move, can't spin. DNA is tethered to all those proteins, it's immobilized, right? So when TF2H starts, using its translocase activity, what that means is it walks along the DNA. So it's trying to walk, say, this way, and the DNA is stuck. What that does is it unwinds the DNA because it's spinning the DNA. So this end is spinning over here. After all the DNA in here, it's all stuck in place, and because one end's spinning and one end is stuck, and as long as you're spinning it in the right way, you're going to unwind the DNA in here. You're going to break those hydrogen bonds. So that's pretty neat. I like that mechanism. And you can imagine it, probably involves ATP. So, okay, so that's how the DNA unwinds, and transcription can begin at the +1 site. Okay, so that's initiation of transcription. So transcription, initiation by pol II at +1 site. Okay, that's what we covered. And you can probably, similar processes are going to be used by pol I and pol III, involving pol I and pol III and their general transcription factors that they need to initiate transcription on their promoters. So because so many of you are going to medical school or applying to medical school and very interested in medicine, I am going to introduce a genetic disease that is related to a defect in one of the general transcription factors. Okay, this disorder is called Spino cerebellar ataxia. So that's a general class of disorders. And there's one in particular that is due to defects in TATA binding protein. So in general, these are called SCAs. So spinocerebellar ataxias, SCAs. SCA17 is due to defects in TATA binding protein. So essentially these are hereditary. Progressive, meaning they get worse with age. Degenerative, degenerative, and often fatal. And they involve the nervous system, as you can probably tell by the cerebellar up there in the name. So SCA17 is a result of mutations in the TATA binding protein, so TBP, which forms part of TF2D. So if we should look at TATA binding protein, the structure, so if this represents the protein with the N-terminal end and the C-terminal end, it's 319 amino acids. And up here, there is a stretch of glutamines. So a stretch of glutamine amino acids. And the normal range for a functional TATA binding protein, TBP, is 30 to 42 glutamines in that region. If we go above 42, well, that's when the disease SCA17 starts. So essentially, and you may, one of the mechanisms that can lead to sort of an expansion of the glutamines here is replication slippage, right? So if we look at the gene here, what we're going to see is lots of CAG codons over and over and over again in this region. And we saw in the past how replication slippage, during DNA replication, can cause an expansion of repetitive DNA. So in this case, we have CAGs and they're repeated, and replication slippage can lead to the expansion of these. And then if we get more than 42 of these codons, well, then that's an allele of the gene that encodes CBP that causes SCA17. So there are two hypotheses to explain why too many glutamines cause problems. And one hypothesis is that, say, proteins with lots of glutamines, where Q is the symbol for glutamine, see, these trigger cell death, or they're toxic. Trigger cell death or toxic. I shouldn't say trigger cell death, they kill the cell. That's probably better. Kill cell because they're toxic. So that's one hypothesis. But there is some evidence to suggest is that TATA binding proteins with too many Qs, too many glutamines, don't function properly in transcription like they're supposed to. And for some reason, which isn't quite understood yet, is that, say, nervous tissue or the nervous system is one of the first systems that, or is a system that is maybe most vulnerable to defects in regulation of transcription. So two competing hypotheses to explain why defects in TBP lead to SCA17. But I thought I'd introduce that because I know many of you are interested in medical school, and so we'll connect some of these mechanisms that we're talking about in this class to genetic disorders in humans. Okay, so that's it for this lecture, and